Hello, I'm Nidhi Rao from Christ Deemed to be University, and today I will be explaining my analysis of cisplatin-treated CHA cells versus control samples for deciphering a biological alteration. So the contents are, I will first introduce cancer and CHA cells, and then I will explain my analysis through MetaboAnalyst, Reactome, Metascape, use of string, and my use of human protein atlas for pathology of cells, along with a literature review and final note. So cancer is basically caused by endogenous agents or exogenous agents, which leads to DNA damage, which can lead to different types of epigenetic alterations, somatic mutations, germline mutations, etc., which basically leads to a deficiency in the DNA repair. So when there's an increase in the deficiency of DNA repair, that is when progression to cancer occurs. So what is cervical cancer? Cervical cancer is basically the cancer of the cervix, which is the part between the uterus and the vaginal canal in the female reproductive system. It is caused by HPV or human papilloma virus. It is the fourth most commonly diagnosed cancer in the world. So this is basically how HPV is transmitted. It is a sexually transmitted virus, and there are more than 100 types of HPV. 15 of these have been identified as putting women at high risk for cervical cancer. So the way it enters and causes infection is this virus enters the cervix through microabrasions or tiny gashes made on the skin, after which it takes several weeks for the HPV to replicate and the infection spreads. 90% of such cases actually heal within 2 years. And of these, 0.8% of cases develop cancer. And 10 to 30 years later, HPV can invade deeper layers of tissues and turn cancerous. There are many different symptoms of cervical cancer. In the beginning stages, cervical cancer can actually be asymptomatic. So later symptoms include vaginal bleeding, contact bleeding, vaginal masses, or pain during sexual intercourse, unusual vaginal discharge, etc. There are some more symptoms of advanced cervical cancer like loss of appetite, fatigue, pelvic pain, back pain, heavy bleeding, bone fractures, and rarely also leakage of urine or feces from the vagina. We must be cancer aware, and women of many ages can actually take the HPV vaccine, which is present, to avoid getting this disease. So there are 3 different types of cervical cancer. The first type is squamous cell carcinoma, which is found in up to 90% of all the cervical cancer cases. It actually affects the squamous cells of the cervix. The second type is adenocarcinoma, which forms in the cells that produces the mucus of the cervix. Cervical mucus is quite essential in fertilization and copulation, etc. Mixed carcinoma, this type has features of the other 2 types of cervical cancer that I just mentioned. So basically, CHA cells are actually a cell line which was derived from a woman who had got cancer in the cervix from Japan. So this is a hypertriploid human cell line with the model chromosome number 71. It contains 1 or 2 integrated copies of HPV16. As you can see, these are the CHA cells here. So cisplatin is actually a chemotherapy medication which was synthesized by human beings. Its function was not known for a very long time, and then people realized that it was a very good chemotherapy drug. So as I have given here, it is given by injection into a vein. It is actually an alkylating reagent, and these agents are most active in the resting phase of the cell. So these drugs are actually non-specific in the cell cycle. Let's get to the data analysis part that I did. The data I received was in a .csv file format, and it contained raw max-quant analyzed mass spectrometry data of CHA controls and cisplatin-treated cells. The data was actually grouped into 2, one for control CHA and another for cisplatin-treated CHA cells. Control group had 4 samples and cisplatin-treated group had 5 samples. The format of the file was P into N matrix, where P equals uniprot IDs of proteins and N was equal to the number of samples. There were different pre-processing requirements like data filtering, missing value imputation, normalization, etc., after which statistical analysis were performed. I used MetaboAnalyst to do the same. So experimental motives, I had a few experimental motives before starting this analysis. I wanted to find the correlation between the given samples. I wanted to find the number of significantly dysregulating proteins or actually quantify the proteins. I wanted to find what the significantly dysregulating proteins were or qualify which proteins they were. I wanted to list out these dysregulating proteins and observe the pathways through which these proteins were involved in. I also wanted to find the effectiveness of cisplatin as a chemotherapy agent or drug. And in the process, I obviously had to analyze various types of cells in cervical cancer and the pathology of cervical cancer. So this was the data I got to analyze, and there was a small correction required. In sample 2, it said CHA control, while the rest of them said control. So I needed to change the labeling to make them all in the same group. And that is what I did in the next slide. Yeah, as you can see, I just changed that. So I used different research processes and analysis processes, such as data filtering and missing value imputation was done on MetaboAnalyst along with normalization. So I also subjected these results to statistical tests on MetaboAnalyst. I uploaded these results to Reactome, and what pathways I found were determined through that. I also used Metascape for some extra information. I used orange for personal interest. I did not add it in this presentation or in the report that I have submitted. HPA was actually used for cell pathology and more information to compare between different types of cancers and mostly the clinical parts. I also used string to check the protein relations that I could find. Then I did a literature review, obviously, so that I could provide inputs on the results of the data that I obtained. So in MetaboAnalyst, I decided to go for one-factor statistical analysis, and I uploaded my data in a .csv format, and I knew that the data type was peak intensities. So this was the format that I used, and I saw that 25.1% missing values were detected in this data processing information. So with data filtering, I used interquartile range filtering. And for missing value estimation, I removed features with greater than 50% missing values, and I estimated these missing values using KNN or k-nearest neighbor method. So I wanted to normalize very well. So I used sample normalization and normalization by median, and I data transformed using log transformation. I did not scale the data. This was the intensity I got before normalization, and this was the intensity that I got after normalization. So these are the sample views before and after normalization. So I then subjected my data to a t-test analysis where I've made the p-value threshold 0.05. I did this because I wanted the test to be very stringent. So I found 520 significant proteins through this method. Then I subjected my data to fold change analysis, where the fold change threshold I gave was 2, and the analysis type was unpaired, and the direction of comparison that I gave was control versus cis-treated. So I found 109 significant downregulated proteins and 92 significant upregulated proteins. Then I went for the volcano plot analysis, where I again put the p-value threshold as 0.05 and raw data, and the group variance I said was equal. So there were 77 significant upregulated proteins and 68 significant downregulated proteins. These were the different proteins that I observed, and I used this as it was very interesting. I uploaded all these proteins onto HPA and checked more as it was intriguing to me, and I wanted to know more. So then I decided to go for PCA, and this is the scree plot for the same. So as you can see, the first PC or principal component has higher variance compared to the next 4. The green line here shows the accumulated variance, and the blue line shows the variance explained by the individual principal component. So this is the PCA plot, 2D score plot that I got. And this was the first component, and this was the second component. As you can see, proper clustering has been observed, and there were no outliers detected, as I had 5 cis-treated samples and 4 control samples. This is a heat map for the entire set of proteins that I got. Here we can see that the cis-treated is shown in pink and the control is shown in green. In the cis-treated area, there are more upregulated proteins on this end and downregulated proteins on this end. And it's the exact opposite for the control. So this heat map is for the top 25 proteins, and this explains the difference between which proteins are actually significantly upregulating and significantly downregulating in a comparison in both like types of samples. So here we can see which ones are upregulated and which ones are downregulated. It's a very beautiful way of observing our proteins. And this was my PLSDA VIP scores. So these are the top 15 proteins with their VIP scores that are given here. And I see that P02671 has the highest VIP score, which means that it is a highly differentiating protein. It is upregulated in cis-treated and downregulated in control, as we can see here. And this P01241 is downregulated in cis-treated and upregulated in control. So we can observe these interesting facts about our proteins. And the dendrogram basically explains the correlation between the given samples. So here, treatment cohorts 6 and 7 are closely related to each other, and treatment cohorts 8 and 9 are closely related to each other. Sample 5 is actually the most distinct treatment cohort, and sample 4 is the most distinct control. So these green ones are the control, and these red ones are the treatment cohorts, as I mentioned. Then I uploaded this data that I got from MetaBioAnalyst to Reactome. And these are all the pathways that I got. The first one being eukaryotic translation elongation, the second being peptide chain elongation, and the third being formation of a pool of free 40S subunits. So this is the first important pathway, and I had 6 submitted entities, which map to 6 Reactome entities. Since I input my data in UniProt IDs, it matches the UniProt IDs by itself. And this is the second important pathway, which is peptide chain elongation. So I inputted 5 submitted entities, and they map to 5 Reactome entities, as I mentioned. And this is the third important pathway, which is formation of a pool of free 40S subunits. So I input these 5 entities, and they map to 5 Reactome entities. So again, I took this data to Metascape, and I checked the analysis. These are the downregulated biological processes that were observed. The most downregulated biological process was cellular response to stress, which is quite characteristic of any type of cancer, as we can see. And these are the other different biological processes, such as response to metal ion, regulation of I-kappa B kinase, NF-kappa B signaling, regeneration, etc. So then I did a HPA search, where I took 5 significantly dysregulated proteins that I was more interested in. And none of these proteins were actually prognostic for cervical cancer, although they showed varying levels of RNA expression. So their protein expressions were also quite varied, and Q01650 was most highly expressed, and hence I took this into further analysis with literature review and string. And the interesting part was almost all of these proteins showed low cancer specificity. That meant that all of these proteins were already present in many different types of cancers, and they don't have any specific identity to a certain cancer. So in string analysis, I realized that the Q16050 protein actually is coded by SLC7A5 gene, and these are the different pathways that it is connected into different other genes. So SLC7A5 actually is a large amino acid transporter small subunit 1, and it basically acts as an amino acid exchanger, and these are its predicted functional partners, as you can see. And string has rightfully observed that I have uploaded Homo sapiens as my current organism. So in my literature analysis, I went through a lot of papers, and these different papers gave me more ideas about how LAT1 was related to different types of cancers. So this one was the most interesting one that I found, as it actually showed that cisplatin can actually increase, cisplatin and its efficiency can actually be increased if we suppress the LAT1 transporter. So I read this in more detail. And as you can see, this is the structure of LAT1. And the CD98 light subunit protein encoded by the SLC7A5 gene was what we were actually looking at. There is another heavy subunit protein, which is encoded by SLC3A2 as well. So basically, studies have shown that LAT1 repression is linked to increased activity of cisplatin in head and neck cancer. And this actually gives us hope to use lesser cisplatin while using LAT1 as a target for different chemotherapy drugs in cancer. So as cisplatin has many different side effects, it will be better if we use lesser amounts of cisplatin and have better efficiency at killing the cancer cells. So hence we can see that the use of proteomics for studying cancer and in cancer biology has been a great benefit to mankind. Thank you very much.